All right, we're gonna just start with a little review of the nervous system to kind of refresh everybody's memory here. So when we look at the nervous system, how it's kind of broken down into just different parts. We have the central nervous system and the peripheral nervous system. And the central nervous system, remember, is made up of the brain and spinal cord. So that's where all the processing occurs in the nervous system. So information is coming into the spinal cord, up the spinal cord to the brain, or it's coming via nerves directly to the brain through the cranial nerves, which we looked at in lab. So brain and spinal cord are the processing areas. There's some instances where the spinal cord does all the processing and elicits the response without the brain. And what do we call that instant response? Going into the spinal cord and right back out. And it causes a response to smooth muscle, skeletal muscle, glands, what?
Reflex.
Yeah, a reflex. So that automatic instantaneous response at the spinal cord level we call a reflex. The brain later becomes aware of it, but it's not required to get that response. So brain and spinal cord are the main regions where information is going in and coming out. For example, when you use your brain to say, I want to move my pencil, the action potential is originating in the neurons of the brain that's traveling down the spinal cord out through the nerves to the arm. So central nervous system, brain and spinal cord. Peripheral nervous system, it's all about the nerves. The nerves are carrying information to and from the spinal cord or the brain, if you talk about the cranial nerves. So some of those nerves are mixed nerves, especially all the spinal nerves that come off of the spinal cord. They're all mixed, which means if I do a cross-section and look at the neurons inside those nerves, I will see axons carrying information to the spinal cord, which are sensory neurons, and then I'll see ones that are traveling away from the spinal cord, which are motor neurons. So when you think of sensory neurons, think of incoming information. Another name for that is afferent information, AFFERENT, so afferent. So information coming, so you got the spinal cord, information coming in. Don't like dried out markers. Information coming in is sensory, and information going out is motor. So motor is going to cause some type of response, right? In terms of skeletal muscle, it's movement. So whenever you think of motor in the voluntary skeletal response, that's some type of movement is involved, okay? So sensory and motor are the two directions of the peripheral nervous system. And then if we look at the motor response, if it's going to voluntary skeletal muscle, those neurons are called somatic motor neurons. Somatic means voluntary skeletal muscle. And then autonomic is anything that's involuntary. So smooth muscle of the digestive tract, the iris of the eye. We looked at that in lab, right? That was our autonomic reflex that you looked at in lab. So that's smooth muscle, which is the iris. And then glands, endocrine glands, that's autonomic. And then the cardiac muscle, because we can't control our heart muscle. So that's the autonomic nervous system. And the way I like to remember that is autonomic, think automatic. You don't have to think about it. It just happens on its own. It's what maintains homeostasis. So those are the responses, again, from the nervous system, from the central nervous system, whether it's voluntary or involuntary. Voluntary, we say somatic nervous system, involuntary, autonomic. And then the autonomic is further broken down, remember, into the rest and digest system, which we call parasympathetic. So that those motor neurons will slow the heart rate, promote contraction of smooth muscle in the digestive tract, right? Promote urination, promote defecation, having a bowel movement, all those rest and digest activities. Where the sympathetic nervous system, the fight or flight system, that's the stress response. So I always think of S, it starts with an S, and it's all about stress, preparing the body to deal with a stressful event, like running from a bear or fighting a bear. But a lot of our stress in today's society, because we live around bears, is just emotional stress, right? So the sympathetic nervous system is still responding to the stress in our lives, but it's not physical stress, it's more emotional, situational stress. So keep this map kind of in mind, because there's a lot of terminology that we're just going to kind of assume that you have that
kind of mapped out. Again, somatic is skeletal muscle, and autonomic is anything that's involuntary. So you could even write next to it, you know, I would write next to it right here, smooth muscle, cardiac muscle, and glands. Write that right next to autonomic. And then next to somatic, you would write skeletal muscle. So right here. And then for motor neurons, that's outgoing information. You could write outgoing. And sensory, you would say incoming information. And that's just a little review of the overarching relationships among the different divisions and branches of the nervous system. So then we have the functional and structural unit of the nervous system, and this is the neuron. So this is where electrical impulses are generated. So remembering the dendrites are the input part of the neuron. So information comes to the neuron through the dendrites. Think of them like antennae. So I would draw arrows going into these dendrites, so you remember that's where information is coming in to the neuron. And there's special types of receptors on the dendrites. We have our sensory receptors, or we have ligand-gated channels, which means some chemical has to bind to them to get a response. So out here at the dendrites, we're going to have sensory receptors. And what are the different sensations? Light, touch, taste, temperature, sound, smell, right? So all those sensory receptors start on the dendrites of those neurons. So we have sensory receptors, those are special channels that open when the specific sense is there. And then we also have ligand-gated, L-I-G-A-N-D, ligand-gated channels, which require a chemical to bind, okay? So they require some neurotransmitter or some chemical to bind to it in order to open up that channel and let ions flow. So the dendrites are all about stimulating the neuron and opening up some channels and letting ions in. And do you remember what ion is found in higher concentration outside of all of our cells? Because of the sodium-potassium pump, what ion do we find higher in concentration outside cells? Sodium. So whenever I open up a sensory receptor, in most cases there's an exception in the special senses, which we'll get to later in the class, but most cases, when we open up a channel at the dendrites, we're going to let sodium flow in because it's higher in concentration outside the cell than inside. So sodium flows in. And then once we get enough sodium in, we get to a special spot in the cell. So this is the cell body. The nucleus is where the processing occurs for all this inflow of ion concentration. If we let enough sodium come in, we get to a special spot on the neuron called the axon hillock where voltage-gated channels are found. And then voltage-gated channels are found all along the axon. So voltage-gated channels are found at the axon hillock, which is this widened area where it starts to narrow to become the axon. So you can label that. It's not labeled on this diagram. Label that axon hillock. H-I-L-L-O-C-K. And there's a lot of voltage-gated channels. So these are channels that only open when there's enough positive charge inside the cell to flip them open. So what's the positive charge? What's flowing in? Sodium. It's positively charged, so that's going to raise that voltage inside that, raise that voltage inside that cell. We'll talk more about that in a minute. Okay, so then this is the axon. So the axon is the highway for information. So its job is just to conduct impulses. So the dendrites receive, and you should label your diagram, with these terms. So the dendrites receive information, axons conduct the information. And the arrow for conduction should always be going away from the cell body in this case. We have a multipolar neuron like this. It's away from the cell body, so it's traveling toward the axon terminal. So you can add an arrow there, going in this direction, right? From cell body to the end. So that's the axon. And what we find on the axon are lots and lots of voltage-gated channels. Like I said, it starts at the axon hillock and then it continues down. So voltage-gated sodium and potassium channels is what we find on the axon. Then we get to the looking at the axon. Some axons are myelinated, so that myelin sheath is an insulator. So the function of the myelin sheath is to insulate the axon and to speed transmission. So any neuron that has axons that are heavily myelinated, they're going to have the fastest transmission of the neurons in the nervous system. So when we look at diseases like multiple sclerosis, where the immune system, it's an autoimmune disease, the immune system attacks its own myelin, then the transmission of those impulses is faulty. And this happens in the brain and spinal cord. So neurons in the brain and spinal cord are demyelinated and causes lots of issues. Weakness, trouble with bowel and bladder function, depression, lots of issues. So there's little breaks though in the myelin you can see. Those are called the nodes of Ranvier, and that's where we see the impulse jump over the myelinated portions. And when an impulse or an action potential forms at the axon hillock, the ion flow happens at these open or bare spots on the neuron, because the insulated areas, nothing can get to those parts of the axon because they're insulated. So it jumps like this from node to node, which is quicker than going all down the full length of the axon. So that's the nodes of Ranvier. And then the cells, the special glial cells you learned about in general A&P that make the myelin sheath are called Schwann cells. So it's kind of like if the axon was a hot dog and the Schwann cell was a hot dog bun, wraps around that segment of the axon and produces myelin underneath it and coats that part of the axon. So where do we see myelination? Only on the axon, right? Because that's where we want good insulation. We don't want to insulate the dendrites because then nothing could stimulate it, right? We don't want to insulate the axon terminals because the axon terminal, that portion of the neuron, its job is to secrete neurotransmitters via secretory vesicles. Do you remember that from general A&P when you looked at maybe the motor end plate and the neuromuscular junction? There's these little vesicles inside those axon terminals that contain neurotransmitters. Do you remember the most common neurotransmitter we talk about in general A&P? Yeah, acetylcholine. So that's made in the cell body, stored in the axon terminals, and then it's released when certain events occur in the neuron to tell it to release those secretory vesicles. So that's what's going to stimulate. So the information, so this releases little secretory vesicles. So you have your neuron with little axon terminals, and it's releasing in most cases, but there's exceptions we're going to talk about. There's lots of other neurotransmitters. We're talking about skeletal muscle with acetylcholine. So that's what gets released from these axon terminals. And that acetylcholine will bind to receptors on the dendrites of the next neuron in series. And again, that would be an example of a ligand-gated channel that's receiving that acetylcholine on the dendrites of the next neuron and then stimulating, an impulse in that neuron. Or that acetylcholine that's released from these axon terminals could be binding to the plasma membrane of a skeletal muscle cell that has receptors for acetylcholine. And then it'll open up that channel, and then sodium again will flow in, right, into the muscle cell because there's more sodium outside the cell than inside the cell. So that's just a little review of neurons and neuron anatomy. Then we'll talk about the action potential. So let's go to our notes before we get into this diagram. So the first slide talks about neurons are highly irritable, which is a description of me before I had my coffee. Most people like coffee. So highly irritable, what that means is that they can change their resting membrane potential, which means they have a certain value, a certain ion concentration inside compared to outside, which gives them a certain voltage. And that can change with a stimulus. Now, all of our cells have a resting membrane potential, but they don't have any way to change that resting membrane potential by opening up channels. But neurons have these special sensory receptors we talked about on the dendrites that can open up channels and change that value. So it just means they can change their value with an adequate stimulus, and then we can get an action potential or nerve impulse. But key concept is it's always the same regardless of the stimulus. So once I get an action potential, it flows down the axon to the axon terminals undisturbed, and it doesn't get any stronger, and we'll talk about that. So these channels in the membrane of our neurons, there's two types. We talked about the gated channels, chemically gated or ligand gated. They open when a specific neurotransmitter binds, or medicine. That's what all of the drugs we give in the hospital, in the clinic, and at home. Those are binding to channels and either promoting a response or blocking a response by that cell. So we're just giving those failed organs and homeostatic mechanisms a little boost. And then again, voltage gated. They open and change, or open and close in response to a change in membrane potential, which means ions are flowing, sodium or potassium, or other ions for that matter. And then mechanically gated. Mechanically gated means we physically deform the membrane. So deform it means when you touch your skin, you open up mechanically gated channels. So just the morphing of the membrane on your skin opens up channels, allows sodium to flow enough that it sends an action potential and goes all the way up to your brain to tell you that you touched your skin, or somebody's tapping you, or you have a mosquito crawling or biting your skin. So those are all examples of mechanically gated channels. So when you look at the resting membrane potential of a neuron, it's at -70. And where that comes from, that -70 inside the cell, so the inside of the cell is at a value of -70 millivolts based on the ions inside compared to the ion concentration outside. So inside the cell is -70. And some factors that relate to that is we have leak channels on the neuron, but there's a lot more ion channels, a lot more leak channels for potassium compared to sodium. So we see more diffusion of potassium out than sodium in. So if both of them are positively charged, if we're losing positive ions faster than we're gaining, it's going to leave us with a negative inside, because there's something inside that's negatively charged, and that's proteins. Proteins made by the cell, all cells, most cells make proteins, neurons in particular. They produce these neurotransmitters, many of which are proteins. So we have these negatively charged proteins that are trapped inside the cell. They're too big to spit through these tiny little membrane ion channels, so they have to leave by exocytosis. So they're stuck inside the cell, making it negative in there. So we have these negatively charged proteins, and then we have this loss of potassium through these leak channels. And the reason why potassium leaves is because of the action of the sodium-potassium pump. Do you remember what the sodium-potassium pump does? So the sodium-potassium pump moves ions against their concentration gradient. So this is the pump. So we see there's lots of sodium outside the cell, right? Lots of potassium inside the cell. So if I opened up a leak channel for potassium, which way would potassium leak? Into the cell or out of the cell? Here's the inside of the cell here. It would leak out because there's higher concentration here than there is here. And if I opened up a leak channel for sodium, which way would sodium flow? In, because there's so much sodium out here outside the cell. So what the sodium-potassium pump does, because it's an active transport pump, it requires ATP. It's pumping against the concentration gradient. So it's going to add more sodium. It's going to take sodium and take it from low concentration to high concentration. So it's going to take this sodium here, this little bit that's in here, and it's going to dump it right back out again. And it's going to take this potassium and bring that in. And because of that, it's establishing these concentration gradients, isn't it? That's where this high sodium outside versus high potassium inside came from, is because the pump is doing that. It's kind of like bailing water that keeps flowing into a leaky boat. It just keeps pumping it right back out again. So there's 2 binding sites for potassium and 3 for sodium. And again, ATP is required in the process. So here we can see it phosphorylates that pump, drops off that phosphate, and then that's going to energize the pump to take sodium out. And the sodium potassium pump is constantly cycling in all of our cells. Because what would happen if we didn't have a sodium potassium pump? If we have leak channels for sodium and potassium, what would happen eventually? Yeah, it would establish equilibrium and there'd be no movement. So would our neurons be irritable then? Would they be able to change that value? No, there would be no value. There would be no -70. So this pump is constantly moving ions across the membrane against their concentration gradient, which is a good thing. We need to have this high sodium outside, high potassium inside. So when I stimulate the membrane and open up extra channels, we see a movement, we see flow. So when I stimulate, again, sensory receptors that are only specific to sodium at first, more sodium flows into the cell, and what happens to my action potential graph? If I'm sitting out here at -70, like in this diagram here, -70 is the resting membrane potential, and that was established by the sodium-potassium pump, negatively charged proteins, and all these extra leak channels for potassium, which is causing more potassium to leave than positively charged sodium to come in. We have this -70 value. Now I apply a stimulus to it. So I stimulate the dendrites, which in most cases open up sodium channels. What happens to that -70 if I bring in positive sodium through special channels? It increases, it becomes less negative, right? Because I'm adding positively charged sodium ions. So instead of being at -70, I start to climb upward. And that's what I see here. I'm starting to climb upward. So what I'm doing is I'm letting sodium here to come into my dendrites. I'm stimulating the dendrites with sound, temperature, touch, whatever. And sodium is flowing in. It's becoming more positive inside the cell. But then if I don't continue to stimulate, what happens? Sodium potassium pump cleans everything up, channels shut, and I'm back down to resting membrane potential. So that's what these are, just failed stimuli that didn't get to threshold. Threshold is -55. So there's a special name for these potentials. They're called graded potentials. We're going to talk about that in a minute. So these stimuli keep coming in, failing, and then I don't quite reach threshold. But if I let enough sodium in to get to -55, that flow of sodium spread throughout the cell reaches the axon hillock. And what did we say? What type of channels do we have at the axon hillock? Voltage gated channels. So once we get to -55, these channels flip open, and they flip open in series all the way down the neuron, following this action potential graph. Whoops. So once I reach threshold, all the voltage gated channels on the axon hillock open up, and I see a rapid rush of sodium into the cell until it reaches +40 inside my neuron. And at +40, sodium channels are signaled to shut. They do not remain open beyond +40. And then potassium channels that are on that membrane, voltage gated potassium channels, they're stimulated to open. In which way does potassium want to flow based on its concentration gradient? Out of the cell. So once I open up extra potassium channels, I see a rapid loss of potassium, which is positively charged. So what happens to my charge inside the cell if I start losing positive ion? It drops back down again, right? And it drops back down until I get all the way to -70, and all the sodium channels were already closed because they shut at +40. Now all my potassium channels close at -70, and I'm back to the resting state again. What this graph doesn't show, until I slide it down a little bit, we see a little bit of this. This is called hyperpolarization because it's a little lower than resting, and that's because potassium channels are slowly closing. So as they're signaled to close, a little more potassium continues to leak out to make it lower than -70. But then at this point, they're all shut. And what's cleaning everything up again, putting everything back where it belongs? Yep, the sodium potassium pump. So if you want to label this little part, this graph right here, this is the sodium potassium pump cleaning everything up and putting it back to its resting state until the next impulse comes along. So this depolarization is happening here to start the impulse at the axon hillock, and it travels down the axon. So this first depolarization occurs here, and we have rapid depolarization followed by repolarization at every bare spot on the axon. At the axon hillock is where all this action is occurring. Until it reaches the end, then we release our neurotransmitter, or whatever the stimulus is for the next organ in the series, or neuron. So in order to get what we call an action potential, an action potential involves those voltage-gated channels on the axon hillock and the axon. That's only going to happen if we hit -55. So and nothing is actually moving down the axon. It's kind of like if you're doing the wave in a stadium at a football game. You stand up, right? And when you stand up, it causes the person next to you to stand up and sit down, which causes the next one. And then all of a sudden you look across the stadium, you see this rolling view, right? Going around the stadium. It's pretty cool. But did anybody leave their seat? No. It's just, think of the people that are standing up and sitting down like sodium channels and potassium channels moving around the membrane, generating an impulse, but nothing actually moves other than ions in and out of the cell at that particular spot on the axon. So that's really important. And that's why when people have electrolyte imbalances, sodium, potassium levels are off, we see lots of issues. They have sometimes weakness, sometimes they have heart arrhythmias, sometimes they're really twitchy and they're like just jerking, laying there really lethargic mentally, but their muscles are just twitching. We need to correct those imbalances very quickly, and that's why we know if you have a stomach virus and you're losing ions through vomiting and diarrhea, it's important to replace those with, like Gatorade or Pedialyte if it's a child. Okay, so looking back then, we'll go back to our notes here. I can't remember which one I put my notes on. It's not that one. Oh my goodness. Here, okay. So there's two types of signals on neurons. Graded potentials occur at the dendrites. So they occur at the dendrites, and the channels involved in graded potentials are those early stimulations that may not reach threshold. So they fizzle out, like it shows in our graph where it says failed initiations on that graph I showed you a little bit ago. And they can go in either direction on the membrane. But action potentials, those are formed on the axon hillock. They can only go in one direction because once the sodium channels close, potassium opens, so we can't go backwards. We have to keep moving in a forward direction down the axon. And we only see those in neuron and muscle cells. So these are the cells that are irritable. They can change their resting membrane potential. And it's an all or none. Either it reaches threshold or it doesn't. If it doesn't reach threshold, it fizzles out and is gone. And does that, am I aware of that then in my brain? Does that travel then if it fizzles out? No. So we have lots of little stimuli occurring in our cells, but if it doesn't reach threshold, it doesn't make the trip. So graded potentials can either go up or down. So if it's a hyperpolarization, what ions are opening up? What happens to the cell in a hyperpolarization? Does it become more positive or more negative inside? If you look at your graph here, it becomes more negative. So what have to open up? What ion channels would have to open up to make it more negative inside the cell? Potassium, yeah. If we opened up channels for potassium right off the bat, We know that there's lots of potassium inside the cell because of the pump, right? The pump keeps pumping it in there. So if I open up a channel, which way does potassium always want to flow? Out of the cell. And potassium is positively charged. So if it flows out of the cell, it's going to be more negative inside. So any channel, any stimulus that opens up a potassium channel, it's going to hyperpolarize our membrane. It's going to go more negative. What negatively charged ion do we find in the extracellular fluid, found more outside of our cells? It's paired with sodium in our diet. Chloride. Chloride ion is a negatively charged ion found in higher concentration outside of our cells. So if I open up a chloride ion with some stimulus, that's going to hyperpolarize my cell, wouldn't it? Because chloride ion would want to go into the cell because it's in higher concentration outside. So if I open up a channel, it's going to flow in. If I bring negative charge into the cell, bam, I'm dropping my resting membrane potential again. So would that cell be harder to stimulate or easier to stimulate if I hyperpolarized it? Harder, because I'm taking it further from threshold, aren't I? I'm down here. Threshold is up here. So if I cause a stimulus that opens up potassium or chloride channels, I'm going in the wrong direction to get to threshold. So this is inhibiting my cell. It's inhibiting it. We call that an inhibitory postsynaptic potential. It's a type of graded potential that hyperpolarizes. And if I open up a channel for sodium or calcium, that would those are in higher concentration outside the cell, so those would flow in and make my cell more positive. So those are all excitatory types of stimuli, and that would bring us closer to threshold. So these graded potentials can go in either direction. Oh, I did it again. I have no memory today. Okay. So they can be depolarizations or hyperpolarizations. Again, if it's a depolarization, it's going to excite the cell, bring it closer to threshold. And if it's a hyperpolarization, it's going to inhibit the cell and bring it further from threshold. So again, this is just a diagram showing that it can go in two different directions because it's not involving potassium channels yet, because those are voltage sensitive. So this is just when I open up some sodium channels, it's going to spread in both directions, and then it's going to fizzle out if I don't continue to stimulate. But an action potential does not fizzle out, so it does not decrease in magnitude. And what is the magnitude of an action potential based on our graph? How tall does it get before it goes down? Plus 40. Yep. And it doesn't get any taller because at plus 40, voltage-gated sodium channels close and voltage-gated potassium channels open. And I can't get any more positive if there's no more positive flow into the cell, right? And if I open up positive potassium channels, that's going to flow out. And that's going to bring us back down to the negative values. So cannot get any taller. So we already talked about resting membrane potential. Threshold is negative 55 as it's climbing up to plus, this says plus 30. Depends on your textbook, plus 30, plus 40. I'm not going to hold you accountable for that specific number. Or if I do, it'll be either plus 30 or plus 40, and you'll know. There won't be two choices. And then again, this is that hyperpolarization because of slow closing potassium channels, and then the action of the sodium potassium pump is cleaning things up and bringing it back to resting. So how do I get an increasing stimulus? What is a whisper versus a shout in my ear? Well, here's the graph of action potential. So each one of these sharp blue lines here represents an action potential. So here I have some action potentials kind of spread apart. Here I have a bunch of them really close together. So there's high frequency of action potentials here, right? I have a lot of them right after one another. And these I've got a little space in between, and here I don't have any. So in here, this is showing the stimulus. Here, this stimulus, so I had a little graded potential. I had some positive charge inside my cell, but it didn't reach threshold, right? It's below threshold. So did I get action potentials here? Nope, it was a sub-threshold stimulus. Now this one reached threshold. Not quite as high, so not quite as much sodium flow. And look at the frequency of action potentials. Here I have a greater stimulus, so more, like this would be a loud sound versus a quieter sound, or a bright light versus soft light, you know, any stimulus. Spicy food versus mild food, right? Hot salsa versus mild salsa. So I've got fewer action potentials here. Are these action potentials higher though, or any one higher than the other? No, plus 30 is the max, but I have more action potentials. So the key concept is more stimulus, more frequent action potentials. So if I have a higher stimulus, so loud sound means more action potentials. Quieter sound, fewer action potentials, not taller. The only way we get taller graphs is looking at those graded potentials. They either go up close to the threshold, like maybe as far as -50, but then they fizzle out. Or some might go as high as -25, and then they fizzle out. So are those differing in height? -50 versus -25, yeah, that's different magnitude. But once it reaches threshold, bam, we're off and running. It's like a horse race or a dog race. Once that gun fires, the gates open and the horses or dogs run around the track. You can't stop it. Questions on that? So this is a nice little table from your textbook that compares the graded potential versus the action potential. And there'll be a lot of questions on this first test about that. So where do graded potentials form? Cell body and dendrites. That's where those special receptors are located to let a little bit of sodium or potassium or whatever ion in. And at the axon hillock and the axon are where the voltage gated channels are. So these can travel the whole distance down the neuron. The action potentials are the long distance signals. These fizzle out on the cell body. Again, these can get a little taller, but always less than -55. And these go up to plus 30 or 40, and then they come back down. They're all the same size, same height. And the stimulus for opening is either chemical or some sensory receptor stimulus. And these are just voltage gated. So once they get to action potential, or once they get to threshold, we get that action potential. And positive feedback cycle means that, again, it travels down the axon uninhibited. It just goes right down. That's because when I open up one part of the axon and sodium flows in, that's going to stimulate threshold in the next part of the axon, which is going to stimulate threshold in the next part. It's going to continue down along the axon. So some sodium flowing in causes more sodium, which causes more sodium, which causes more sodium. That's an example of positive feedback, right? When a stimulus causes more of the same thing. We don't see that with graded potentials. They come in, they either reach threshold or they fizzle out, unless we keep stimulating the membrane. And then repolarization, that occurs, right, when we reach plus 30 or 40 on the membrane, and then that opens up potassium channels. Well, these never repolarize because they just fizzle out. They never get to the plus 40 or 30 to stimulate those voltage-gated channels. So when we look at the two types of graded potentials, which I was talking about, we have the excitatory, which we can abbreviate as EPSP. That means I'm depolarizing closer to threshold, and inhibitory, or IPSP, is taking me further from threshold. So what examples did I give again? See if you can generate it now. What ion channels would open to give me an EPSP, which is a graded potential that's going closer to threshold? Sodium. What? Yeah, very good. Sodium or calcium. So you could write that down underneath EPSP. Sodium or calcium channels, if they open on the dendrites and cell body, right? If those open, that's a graded potential that's going closer to threshold. So that's an EPSP. It's exciting my membrane. But if I open up a channel that hyperpolarizes my membrane, takes me further from threshold, that's going to inhibit my membrane, and I'm going to be further from threshold. And what ion channels would cause an IPSP? Potassium, because it's in higher concentration in, so it's going to flow out. And what's another one? Chloride ion, right? Because that's negatively charged, and if that flows into my cell, based on high to low concentration difference, that's going to hyperpolarize. So some of our medicines do that. They encourage inhibition of our membranes. And what major sensation are we always trying to inhibit among our patients in the hospital setting? Pain. Very good. Pain. So we're trying to shut down and make those cells less responsive. And the problem with our pain medicines now, though, we don't do a great job on treating the pain at the source. We do nerve blocks, right? And those numb the pain for a period of time, but then they wear out. And the physician is not going to keep coming in your room and giving you nerve blocks, right? So what do? We inhibit the brain from perceiving pain. And when we shut down the brain from perceiving pain, we're also shutting down the brain from wanting to have a conversation, wanting to get out of bed, wanting to go to physical therapy, right? What do people do that are on a lot of pain meds? Sleep and sit there, right? Makes for easy patients, right? So it's easy when someone says, I have a lot of pain. What are you rating at 10? Here, take this. We'll give you this. We'll give you this. And I'll give you this by IV. And it's a happy patient, right? Sleeping, passed out, not causing any trouble. But the problem is dead sores, lack of appetite, not participating in physical therapy, not moving. Now you're slowing their recovery because you're shutting down their brain and all those other important areas. So we always try to give the least amount of pain medicine to our patients to keep them active and promoting their recovery, not just covering pain. But now, if it's the day after surgery or the day of surgery, is it okay to sleep and whack them out a little bit? It is, because nothing's going to happen in that first day of surgery. Like on our floor, they crack the chest, they're opening up all, the sternum, it's very, and then they have that chest tube. It's a lot of pain. So our goal is just to get them to walk to the door and back in their room. We can get them to do that, then at least we've got muscles working, blood clot prevention and all that kind of stuff. But we really need to work harder as a society on pain medicines. We've been relying on opioid, which are the pain medicines that make you sleepy and tired. We've been relying on those for a long time, right? People have been using that for hundreds of years. And we need to do better. We need to find other ways to block pain that don't shut down the nervous system. Because what happens is people switch from, yeah, I've got all this pain, and then you give them this medicine, and people with anxiety and just general unrest in their lives, well, they like that sleepy feeling when they're on the pain medicine. Then after the pain goes away and they're starting to heal, they find themselves, I'll just still take some of that pain medicine because it makes me feel sleepy and relaxed and not stressed out, and I just feel really at peace. I mean, doesn't everybody want to feel that way? So that's where the pain medicine turns into an addiction problem. So we always treat with the least amount of pain medicine necessary to treat the problem and not contribute to dependency. And what people say accidentally to me is, yeah, I'll take some more of that fentanyl because I just really need to relax. And, we don't give fentanyl to relax. That's what people do heroin for, right? It's to relax, but that's not where we want to go. So that's when we say, well, the doctor, and what the doctors do to make the nurses' lives easier is they will discontinue those IV pain medicines, so we do not have access to them to even give to the patient. They'll say, oh yeah, the doctor discontinued, now we have Tylenol. They hate that, right? Some people, Tylenol. And sometimes Tylenol is not a good medicine. Me personally, for my little aches and pains and with my kids, ibuprofen seems to work a little better, but older people are at risk for stomach bleeding and lots of issues. So they don't like to give ibuprofen. Anyway, moving on then. So speeding the impulse down the axon, a couple of factors promote the transmission down the axon, and we already talked about myelination, right? If an axon is myelinated, it's going to jump down the axon from node to node to node. And we call that saltatory conduction. Saltatory just means kind of jumping down the membrane. Like if you were trying to run up the steps or run down the steps in a hurry, say there was a fire and you need to get out of this building quickly,
Some of you might skip some steps going down, right, to get down quicker, or heaven forbid you're late to class and you're skipping steps, right, to go upstairs, or you forgot something upstairs in the bedroom at home, you skip steps. So that's the same concept, it's just quicker that way. Axon diameter, the larger the diameter of the fibers, the faster the flow. So we see that those types of neuron fibers where you want, a quick reaction. So our reflexes, the neurons traveling to and from our skeletal muscle, they're heavy myelinated, thick axons for quick conduction. So this just shows the difference in, if I have to go down every single inch of that axon, it's going to take longer than if I can jump down the axon on a myelinated axon. So like I said, multiple sclerosis is an autoimmune disease. It's not, I did my graduate research on this disease, so it's not necessarily accurate to say that it mainly affects young adults. That's the most common time that people are diagnosed, but there have been people that have been diagnosed later in life, and the earlier someone is diagnosed, the better the course of the disease. They tend to have this kind of relapsing, remitting version of the disease where it gets bad for a while and then it goes away. And then they don't think about it. Then it comes back and then it goes away. But every time it comes back, it's a little worse than the previous time. So it's always a kind of a decline over time. But I know several people that were diagnosed in their 20s and they're doing okay. But by the time they get to their 40s and 50s, it goes from this relapsing, remitting course to this progressive course where they start to decline a lot more rapidly. And I know someone who lost their life to multiple sclerosis. So it really depends on the individual, and it's really important to get treatment right away because the medicines for multiple sclerosis prevent the attack on the myelin. So for people that don't like to take their medicines, and a lot of it is injections or IV, and they don't like to take those because who likes to take shots, right? So people like to take the least invasive version. Like I was talking to a checkout girl at a grocery store I used to visit that had MS, and she's like, yeah, I chose to take this medicine because I don't like to give myself shots that often, and this one I don't have to do it as often. But if you look at the effectiveness of that medicine, it's not nearly as effective as the medicines where you need more injections. So what do you want in the course of a lifetime? The inconvenience of injections and protecting your myelin, or less injections but less effective? So you really have to do your research because doctors are going to be fine with saying, oh, we can use this method. It's not as effective. Your quality of life will decline quicker. So we're really going to advocate for ourselves nowadays. So a big concept, though, is it's in the central nervous system. There's two diseases that cause multiple sclerosis, and this would be on an NCLEX exam for sure because they're common, not common, but we see them out there. Multiple sclerosis is demyelination of the brain and spinal cord. I said nothing about nerves, right? Brain and spinal cord. So the neurons in the spinal cord, the neurons in the brain. Another disease called Guillain-Barré, maybe you've heard of that. You don't have to worry about writing this down, but you will hear about it in nursing school. I'm not even sure of the spelling myself, but it's called Guillain-Barré. Kind of looks like Guillain-Barré. That is demyelination of the nerves. And this is something that some people have had a reaction to a virus, and all of a sudden they become paralyzed from the neck down. We've had patients like this. We had a young girl that had it. This comes out of nowhere. We've had elderly patients that have it. All of a sudden they're paralyzed. And then it runs its course, and then slowly they get better. And one girl, she was completely recovered. Another person, it took them a long time, and they didn't fully recover. It just depends on the course of the disease. But some people have had Guillain-Barré when they've had a vaccine because it stimulates the immune response, and whatever the reason, they get an over response to that antigen in the vaccine, and then they end up with this Guillain-Barré. It's very rare, so the risk of not becoming vaccinated and getting, encephalopathy from chickenpox and some of these other complications of the disease that we're trying to prevent with the vaccines, that's more common than this Guillain-Barré, but it is out there and it does happen to some people in rare cases. So we can classify nerves again by their diameter, their degree of myelination, and their speed of conduction. So
We just classify them as type A, type B fibers. So group A fibers, large diameter, myelinated, somatic. What does somatic mean? Skeletal muscle. So these are the fibers going to and from our skeletal muscle. So that would make sense, right? Wouldn't you want them to be fast at what they do? Because we're trying to move around our environment, right? And keep ourselves safe. So skeletal muscle, sensory and motor. So we need lots of input coming in from our muscles and going out to our muscles so we can sit, stand, move around like we need to. And then the group B and C fibers, these are autonomic nervous system fibers. So ANS means autonomic nervous system fibers. And what do those fibers supply? What parts? Okay, what type of cells? Yeah, cardiac muscle, smooth muscle, glands, endocrine glands. So group C and group B fibers are serving those areas. So because of that, that's a good thing, right? Fight or flight, rest and digest. We don't want rapid transmission, right? Because if you're going to stimulate the bowel to contract, we want rapid bowel contraction? No, that would be bad. Or do we want rapid insulin secretion? No, we want that to be more, slow, not like a reflex. So group A fibers really keep us safe because that's skeletal muscle. So when we're looking at trying to desensitize fibers, those group A fibers are the ones that, with pain anyway, are the ones we're trying to decrease the perception of that information coming in. All right, so we have the space between neurons called the synapse. So synapse means space. It's a space between one neuron and the next neuron in series. So a presynaptic neuron is the one that gets the stimulus first, and then it's going to try to stimulate the postsynaptic neuron. So there's a space between those neurons, which we call the synapse. So the presynaptic neuron conducts impulses toward the synapse. So it has the axon terminals, right? It's the one with the axon terminals on one side. And then the postsynaptic neuron has the axon terminal, or has the dendrites in most cases, if we're talking about simple transmission. Okay, so we have our synapse, which is just the space. So here's the axon terminal. This is just one axon terminal. That's why it's so small, right? We know we have multiple axon terminals. So here's one axon terminal. So this would be the presynaptic neuron, right? And then this would be the postsynaptic neuron. So this might be the dendrites or cell body of another neuron that's traveling, up the spinal cord. So there's this space between the two, which we call the synapse. So looking a little closer, what do we know about the axon terminal? What does it have stored inside of the axon terminal? What? Acetylcholine, if it is a specific type of neuron we're going to talk about, that's one of those. And acetylcholine is a neurotransmitter, right? So there's different neurotransmitters stored in the presynaptic neuron that need to be released and bind to the dendrites, to a receptor. What type of receptor would we need if this is a dendrite? What type of receptor would we need here? What do we call those channels? Ligand gated. We would need a ligand gated channel, chemically gated, for whatever neurotransmitters here will bind to a channel here and cause this channel to open. And what ion is floating around in this space outside both of these cells? Sodium, yep, and chloride, right, and calcium. But sodium is the most common ion. So when that opens up, sodium is going to flow in. So let's watch this. So there's those secretory vesicles with neurotransmitters in the presynaptic membrane, right? So they're going to get ready to, there's the action potential causing the release of those neurotransmitters. And those neurotransmitters bind to receptors on the postsynaptic membrane. And when it binds, it didn't show that, but see how the action potential is generated now in this membrane? Because sodium channels, these ligand-gated channels, acetylcholine binds and then sodium flows in. And that's going to cause a graded potential in the next neuron unless it reaches threshold. If we get enough neurotransmitter release, then we're going to reach threshold, right? And then a voltage-gated channels, all those will open up on the axon terminal of this postsynaptic membrane. And then eventually enzymes will come in and break down that acetylcholine. So, go back to our PowerPoint. So there's different types of synapses. Some go from the axon terminal onto the cell body. We call those axosomatic. Some go to the dendrites, like we talked about, axodendritic, and some even go right to the axon of another neuron. So we call those axoaxonic. So there's a number of different types of synapses that form, but these are all chemical because there's a neurotransmitter being released. So in all cases, there's an axon terminal releasing neurotransmitters onto the next neuron. A less common type of synapse is an electrical synapse, and that's where there's not a space between them, but there's these channels that connect the axon terminal of one neuron to the dendrites of another. So the flow is direct. So if I have stimulation of the presynaptic neuron and there's all these sodium ions in there, right, and there's enough to reach threshold, the flow of ions are going to go right into the next neuron because they're tethered together by these gap junctions. So make sure you highlight the word gap junctions, and that's unique to electrical synapses. So communication is rapid, and it can be back and forth because of these doors connecting the two neurons. And I'll show you a picture of that. So here is a diagram showing the two. So here's a chemical synapse. So you have action potential coming down. Calcium has to flow in from the extracellular fluid. That's just a process of how chemical synapses work. You studied this a little bit when you did the neuromuscular junction in general A&P. But the neurotransmitter comes across, binds to these ligand-gated channels, and sodium flows in. So then you get your action potential. So that's a chemical synapse, most common in the body. This is the electrical synapse. See how they're tethered together? Your presynaptic cell and your postsynaptic cell. They have these gap junctions for ions to flow directly. So you don't need to release a neurotransmitter. All you have to do is open up ion channels in this membrane, get an action potential going, and all that sodium and potassium flows right through the door into the next cell. But do we get an action potential? Depends, right? Because it's just ion flow. If enough ion flows into this postsynaptic membrane, yeah, we'll get an action potential. But if it doesn't, then we don't. So we need to get enough stimuli to get enough ion flow for an action potential here and keep the action potentials coming to get the sodium to flow into here, or whatever ion is going to stimulate this postsynaptic membrane. So this is an electrical synapse. And just remember the gap junctions, they're always doorways between two cells. And we're going to see gap junctions again this semester. And they're doorways connecting two cells, allowing ions to flow from one cell directly to another cell. Again, chemical synapses are more common, and you have the axon terminal of one secreting synaptic, neurotransmitter through synaptic vesicles to a receptor on the postsynaptic neuron. And again, those are ligand-gated or chemically gated channels for whatever neurotransmitter is being released from the presynaptic neuron. And the space between the pre and postsynaptic neuron is called the synaptic cleft. So it's just a space, and things diffuse from high to low concentration. So it diffuses from the pre to the post and binds to those receptors. So here we just see it diffusing across, binding to the receptor. But we have to have an enzyme present to break down this neurotransmitter on this postsynaptic membrane, because if it sat there the whole time, it would keep that door open and we have constant stimulation of the membrane. We don't want constant stimulation. If I contract a muscle, we don't want constant muscle contraction. Eventually, we need to relax the muscle, right? So there's enzymes in that synaptic cleft, like acetylcholine esterase, that breaks down acetylcholine, and the two parts of acetylcholine, acetic acid and choline, separate and diffuse away from the space, closing the channel and stopping the stimulation. So without the enzyme, we'd have constant stimulation of the membrane. And I think that might be a test question, so remember that. Enzymes stop the stimulation of the postsynaptic membrane. So in some cases, part of it goes back up into the presynaptic neuron to be used again, and some diffuses away. It just depends on the neurotransmitter, how it's broken down. But you have to have an enzyme to stop this process of binding. So here's my neurotransmitter binding to the postsynaptic neuron. Ions are flowing through. We get a graded potential on the postsynaptic neuron. And then again, an enzyme breaks it down, diffuses away, or it's taken back up. We call that reuptake. If part of the neurotransmitter that's broken down by the enzyme is retaken up into the presynaptic neuron. So again, graded potentials are what's formed on the postsynaptic neuron depending on the amount of neurotransmitter released and how long the neurotransmitter is there before it's broken down by enzymes. And those can be excitatory. And again, what did we say about that? How do we know the difference between EPSP and IPSP? Yeah, so which one is more positive, bringing it closer to threshold? What? Excitatory, very good. So that's going to bring it closer to threshold. It's a depolarization. So you can even write that right after EPSP. Depolarization. And IPSP causes what? Hyperpolarization, making it more negative. So again, that's going to inhibit the cell and it's going to lower the transmission. Lower the chance of transmission. So here we can see the stimulus is applied. It's called an EPSP. These are graded potentials, so they're going to always be less than threshold, but it's making it more likely to reach threshold. If I keep stimulating it, it's going to get taller and taller until it reaches threshold. And these are hyperpolarization, so we're getting further from threshold, so that's why it's in IPSP. We're going lower, further away. So one EPSP cannot get to threshold. We need lots of them. So to summate means you have to add them together. To sum means to add together. So if I have, 10 EPSPs, that might allow enough sodium in to get to that -55 and fire off my action potential. But what happens if I get an EPSP followed by an IPSP? I'm back to 0, right? They kind of cancel each other out. So I need more EPSPs than IPSPs. Does that make sense to get to threshold? So they can add up together or cancel each other out depending on what the stimulus is causing the IPSP or the EPSP. So it's all about reaching threshold or not. So there's different neurotransmitters. Some excite, some inhibit. So, or if I don't have enough neurotransmitter, I'm not going to get enough EPSPs to get to threshold. So integration just means I'm adding up EPSPs and IPSPs, integrating the two different types of stimulation or lack of stimulation. So we'll pick up next time looking at different neurotransmitters.